Today's presentation is Mechanisms of Physiological Changes in Drug Exposure in Cancer Patients. Today's speaker is Oliver Hatley. Oliver is currently a senior research scientist at Certara, where he's part of the translational sciences in the DMPK group within Certara's Simcyp team. He's involved in the development of special populations within the Simcyp population-based simulator. Ollie earned his doctorate from the Center for Applied Pharmacokinetic Research at the University of Manchester in the UK. Ollie, welcome to today's event. I'll now turn it over to him to begin the presentation.
Perfect. Thanks, Suzanne. So today, as it's billed, we're going to go through the cancer population and essentially looking at how we can use PBPK to determine exposure in cancer patients. So in terms of the outline of the talk, we're going to talk about some of those physiological considerations that we have to take into account, the application of those that physiology in cancer in predicting mechanistically within PBPK models and how that affects drug disposition. Throughout the talk, I'm going to give some kind of case studies of some of approved drugs, specifically in the oncology field. And then also, I'm just going to touch on some of the applications going further with the cancer population in terms of looking at using tumor model compartments and trying to predict intracellular concentrations within tumors. So just as some sort of background, as many of you will know, that cancer is a massive area of research and development, and globally it accounts for around $133 billion in terms of spending. If you just look at the graph at the bottom, what I've highlighted is over the last 4 years, kind of number of drugs which have been currently worked on in R&D pipelines. And you can see overall in cancer is one of the biggest in terms of that development pipeline. If we move over to the other graph, what you'll see is that in terms of novel drug applications, what you can see is oncology is the largest in that field across the same kind of 4 years. So the point essentially is there's lots of work going on in terms of R&D in oncology, and essentially what we're going to cover is how we can potentially use PBPK in this kind of research and development. And moving on from that, what you can see here is a publication by a group at Genentech, and they reviewed some regulatory submissions. And essentially the take-home message from this was that overall anti-cancer agents were the main applications which used PBPK in that submission. So as you can see, it's very, very relevant for cancer populations in terms of informing the drug development process and also informing kind of drug labeling. And moving on from that, what you can see on this slide and what's highlighted in the boxes are actually Simcyp's direct impact on novel drug approvals. And what you can see out of the 42 compounds on this list, around 45% of them in their drug labeling guidances have PBPK included in them. So as you can see, it's an important area of research. So in terms of developing oncology PBPK populations, there are other reasons for looking to do this. And in part, that is to do with the nature of the disease, which is cancer, in terms of the kind of drugs which are developed. And some of them obviously have narrow therapeutic windows, and there are also kind of safety concerns with dosing healthy volunteers, which would generally be your first port of call when kind of looking at first kind of clinical data. Also, what is kind of goes hand in hand with the development of cancer agent is accelerated drug approval programs. So looking to get drugs for the market as quickly as possible for patients. So these are key reasons why is an advantage to try and use PBPK as a tool for understanding and developing your compound of interest. Another point which I'll come on to a bit later is the fact that there are differences that you would expect between young, healthy individuals and the older population. And in general, cancer is a disease of the older population. In terms of kind of looking to the future as well, in some regards, and it's a bit of a hot topic in many fields, is looking at the impact of precision dosing. And obviously that comes down to looking at optimizing the efficacy and also taking into account that drugs may have a narrow therapeutic window. So essentially it's looking at personalizing that for individuals and different groups of individuals and ensuring that people are getting the right kind of dose. Another factor to consider when looking at trying to develop oncology populations and their kind of utilization is the instance of co-medication. So obviously when you have people who are very sick, there are a large number of medications that are being taken, and also that will increase the potential for drug-drug interactions. And often this is seen, especially in cancer patients. So in terms of coming up with dosage recommendations, PBPK is a tool that can be used towards that. And in that kind of vein, this is partly why different groups have developed their own PBPK populations. And this has been done, and these references I've provided below are from consortium members who have initially built their own Simcyp PBPK oncology populations based on in-house data sets. So we have data sets from Genentech and Sanofi, and then the final publication is from a group at AstraZeneca. And in terms of this kind of interest, obviously that became very influential in terms of getting it voted in as a wish list item for development within the Simcyp simulator. And in version 17, it was the project that was involved in creating this new cancer population. So one thing to consider when talking about a cancer population is that oncology is very diverse, and obviously based on the figure in the background, what you can see is a large array of cancer types, and they will obviously have differences between them in terms of physiology. So the real challenge that we had in terms of creating a PBPK population was trying to look at what common features and physiological changes that we need to take into account when developing a cancer patient population. I'm just going to switch a bit and talk about a drug which has utilized quite cleverly in its development a lot of PBPK in terms of mitigating some clinical studies. In fact, in this case of the Ibrutinib, they ran 24 simulations to replace or at least advise in absence of clinical studies. And in this case, they followed kind of a best practice approach for using PBPK, so building the initial PBPK model based on in vitro data and in clinical data. And what I'll show you on the next slide is how they've verified their model and used it to look at 2 clinical DDI studies. So in this case, they were looking at ketoconazole as an inhibitor of CYP3A and rifampicin as an inducer. And then they applied this model to untested DDI scenarios, so looking at moderate inhibitors and inducers, and going further, looking at what sort of dose adjustments might be required. So if you look at this figure, which is taken from the FDA summary, what you can see is there are the clinical studies in this turquoisey green color, and the solid line represents an observed DDI. And the dashed lines represent their verification using PBPK. And essentially what they were able to do is verify their model in terms of the ketoconazole interaction and the rifampicin interaction. And then they were able to fill in the gaps for weak, moderate, and strong inhibitors and moderate and strong inducers of 3A4. So what they were able to do is basically fill those gaps. And at the bottom, what they came up with in terms of recommendations, and that went directly into the label in terms of what sort of dose adjustments should be made when co-administered with different inhibitors. So one thing I just wanted to draw out from that review was some of the text which was written down in terms of how this potentially could be developed further looking at oncology. So essentially what they noted is that they use PBPK to look at the drug-drug interactions, and the kind of general recommendation was to look at potential PK differences between healthy individuals, which are used for building and simulating, and oncology subjects, and essentially looking at the impact of age, for example, formulation, and other factors in PBPK, and then what potentially that impact could have in terms of interactions, so 3A4 inhibitor or inducers, and what impact that could have overall in that oncology population. So moving back to kind of our kind of process for developing a cancer population, the schematic at the top highlights the kind of overview of how we handle databases within Simcyp. And essentially what we have is this systems data, which represents the population database, so that all the physiological data, and the drug data. And that is obviously put together within a trial design, and those kind of factors interact and result in predictions using IVIVE and PBPK, and predictions of differences in exposure in that population. In terms of the development of that cancer population, we started with the North European Caucasian population as a baseline, and essentially that was because the majority of data that we had was from Caucasian cancer patients. But essentially what we looked for is different differences in physiology which were seen between healthy individuals and cancer patients. And in the next few slides, I'll discuss some of those. In total, we use a large number of literature sources in order to do this kind of analysis, comparing with healthy subjects. The brief essentially was to look at solid tumor cancers, so we didn't, or at least excluded liquid tumor types where possible, and also because this may potentially affect kind of plasma proteins and other biochemistry in the blood. Subjects with other comorbidities were excluded where possible, and obviously in some cases that's not, because people may develop other diseases in kind of combination with cancer. And the third point is pretty rare anyway, but if possible, there was data from untreated cancer patients, but as I said, in terms of having that information, that would be rare and probably unethical in most cases. So moving back to kind of one of the key parameters for building a population is knowing kind of age distribution, because in the way that we build populations, there are correlations which essentially derive from age and sex relationships. So what I've highlighted here is that distribution of ages in the cancer population. And what you can see, over 50% of the population is over 65. So that's going to have an impact on differences between healthy individuals. We looked at kind of the next set of parameters which we look at when developing virtual individuals, and it's looking at different height-age relationships, for example. And as you would expect, cancer isn't really something which generally affects height, especially in adults, at least. So essentially what we were able to show is that data from cancer patients were captured using this North European Caucasian relationship. And that's similar to how other studies have looked at that information as well. One key difference, though, is in height weight, and obviously there are changes in weight which are associated with cancer. Some of this is down to general weight loss, but also loss of muscle mass, for example. So essentially what you'll see is that there is a different relationship with height to weight. So that is taken into account in this population. Another important parameter is looking at renal function, and this is generally seen that there are patients which have renal dysfunction or insufficiency, which isn't actually correlated with cancer type. Something that's quite interesting and a bit more of a challenge in terms of modeling it can be that serum creatinine levels can appear normal, but creatinine clearance can be still reduced, and that's possibly linked to changes in muscle mass in cancer patients. Another factor, and what we consider within Simcyp, is changes of GFR with age, but that doesn't necessarily explain the more pronounced reductions in GFR in, for example, patients with ovarian cancer. And this really hasn't significant impact, especially for drugs which are renally eliminated. So, for example, there's a publication looking at cisplatin, and a GFR of treated patients is generally lower than age-matched untreated patients. And there are also considerations for drugs which have narrow therapeutic windows. For example, this drug, carboplatin, you can have a AUC between 4 and 6 and see that the drug is effective, but a range of 5 to 7 can increase the incidences of myelotoxicity. And on the kind of flip side of that, any kind of dose reduction as much as 10% can result in doubling of 5-year relapse rate. So in this case, what we're really interested is trying to find that sweet spot in terms of that drug. So in terms of methods which are available for predicting GFR in cancer, the classic ones that we use, especially within Simcyp, is looking at the Cochrane and GALT, and also the modified diet in renal disease. And essentially, there have been studies that have looked at the accuracy of these equations to predict GFR in cancer patients. And generally, there is seen that Cochrane GALT is more accurate, but it's been more appropriate in some circumstances to use alternative equations. So essentially, we've looked at this in terms of studies which have been published, but also looking at our own internal kind of verification process. And what we generally see is that the right formula gives improved predictions over the other kind of 3, as in this study here. But there are kind of caveats, and essentially it only shows improvement over other equations in kind of the normal GFR range. So that's something to be considered. And essentially what we provide within Simcyp is kind of scripting tools which allow for modification of this GFR prediction in order to better capture different patients. Another essential part of kind of predicting exposure is looking at concentrations of plasma proteins. And typically there are changes observed in cancer patients. So albumin is one of the important protein binding proteins in protein for binding of drugs in plasma. Interestingly, for this for albumin, you might not see any changes in the earlier stages of cancer, but as disease progresses, this can drop significantly. In terms of alpha-acid-glycoprotein, this can increase in cancer patients and versus healthy individuals. So what I've shown here is 2 scenarios. So these are measured drugs in cancer patients and in healthy individuals. And what you can see is the reduction of albumin results in a decrease in binding of itraconazole in this case. But a drug such as fluconazole, which binds alpha-acid-glycoprotein, when this obviously increases the amount of bound drug. And that in turn will have impacts on the amount of drug available for example, metabolism. If we look at another drug here, fentanyl, and again this drug binds primarily to alpha-acid-glycoprotein, And what you can see in this poster by my colleague, Nika, she looked at predictions using the Sim-Cancer population of fraction unbound for this, for fentanyl. As you can see, what you get is this reduction in fraction unbound, and the predictions are pretty closely observed. So this slide generalizes how we put all this information together to build the population. So we've talked about the age-height relationships, the weight-height, looking at different levels of those plasma proteins. I haven't discussed, but we take into account differences, for example, in hepatic transporter abundances, which may be seen between healthy individuals and cancer patients, and also looking at serum creatinine and predictions of GFR. So this is some of the verification that we did when building this population. So what does that mean overall? And this figure is a hive plot for those who have not seen this kind of figure before. And essentially, each strand represents an individual, and in this case, we've simulated 1,000 individuals as the North European Caucasian in red, and in blue, the Sim-Cancer population. And what you can do with this kind of plot is visualize differences between different parameters. So here we have age, for example, and there's a higher density of blue, which indicates there's higher age of individuals with these simulations in cancer. We can look at predictions of GFR and kind of ranges as well within different populations. Serum creatinine can be higher in cancer patients, as well as alpha-acid glycoprotein. As we've discussed, there are changes in albumin levels. Going further, we've obviously done verification of this model in terms of the impact on predictions of probe drugs. One of the interesting things to comment on here, and it's still kind of an issue of debate when looking at cancer populations, is any reduction in intrinsic clearance. So, for example, CYP3A substrate midazolam has been reported to be undergo a decrease if you compared cancer patients to healthy individuals. So when we did our initial performance verification, we didn't make any changes to the CYP abundance. And part of the reasons behind this and why we believe this might be relevant is because of the impact of age. And in this case, what we see is that as patients are older, there are changes in cardiac output, but also changes in the liver volume, and that will primarily drive decreases in intrinsic clearance based on that liver volume. So what we were seeing is that there was a reduction in this intrinsic clearance as a result, and in turn this brought predictions of, for example, clearance, and looking at the bottom here, looking at impact on predictions of AUC. So what we're comparing is AUC measured in cancer individuals and AUC measured in healthy individuals. One of the things to show and highlight in this analysis at the bottom is that there is a lot of variability between studies, and in this case there was only one example where there was a matched kind of clinical study. All these are from independent studies, so that can cloud some of the interpretation. but shows generally the predictions are within kind of the observed data that we've seen. Moving on, I've brought out here a few other examples where model-informed oncology drug labeling has been undertaken. So in the first example here, what they were looking at is bridging healthy drug-drug interactions to cancer patients, so similar to what I was talking about previously, or with the example of Ibrutinib, but taking that further into impact of interactions in cancer patients. And in this case, what they showed was a reduction in the DDI in cancer patients. Looking at a second example, in this case, what they were looking at is impacts on absorption and food effects, and how that would be affected in cancer. And the final example here that I've listed is an example where they've looked at bridging to pediatric dose. And essentially what they were looking at, because of the PD effect here in terms of toxicity, what they were trying to use PBPK for was to suggest appropriate doses for different ages. So taking kind of this further, moving on from kind of pharmacokinetics, I just want to briefly talk about pharmacodynamics, and this is something that we've looked at within the oncology population. And the example I'm going to show you now is based on Paclitaxel. And essentially, this is a drug which is a substrate for 3A4 and 2C8, and therefore there are impact and risk for drug-drug interactions, as well as the effect of polymorphisms. So the other thing to consider is that there is this kind of toxicity effect where you get changes in neutrophil levels, and essentially that can be modeled looking at kind of delayed effects after administration of Paclitaxel. And essentially this was done by my colleague Melis. And this schematic kind of shows that kind of process, and in terms of building that model and refining it, looking at drug-drug interactions and performing verification of those parameters within the model, and then applying a PD model to look at the effect of neutrophil counts. And the application is obviously looking at the impact of changing dose, but also any kind of interaction and how that will affect the required dose based on interactions with inhibitors, for example. The other thing I briefly wanted to introduce to you was the tumor distribution model. And we have this available within Simcyp for minimal PBPK modeling, as well as full PBPK modeling. And essentially this is available using a separate compartment receiving blood flow from the systemic compartment. Or in the full PBPK model, you can have it parallel as an additional kind of organ modeled in this way, or you can kind of specify a lung tumor. And the essential difference between these two is the kind of blood flow that is received. So in this case, the lung tumor will receive venous blood, and the systemic tumor will receive arterial blood. Something to point out is this is available in the human simulator in terms of substrates and an inhibitor, as well as the substrate primary metabolite. In terms of animal simulators, we have this available as well within the mouse simulator as substrate and primary metabolite, but because there aren't any interactions which are able to be simulated in the mouse, and there's no extra option for inhibitor. One other thing to mention is that we can consider tumor growth as well as shrinkage, and that can be modeled dynamically in terms of pre-treatment growth, as well as the effect of treatment on that tumor, and also kind of changes within the tumor can be modeled. So, for example, changes in blood flow, for example, or different compositions of that tumor can be modeled as well. Kind of how that works out is this is the kind of structure of the model, and essentially it's a permeability limited distribution model, and that is based on the transfer of unbound unionized drug. One assumption that's made is that there is instantaneous equilibrium between the vascular space and extracellular water, and that's considered reasonable because tumor can be leaky. It's also possible to take into account passive and active transfer between the extracellular water and intracellular water, sorry. And within that, you'll have impact of, for example, transporters, so we can take into account, for example, PGP, or other transporters can be modeled as well. Within the intracellular space, you can get binding to neutral phospholipids, neutrolipids, and acidic phospholipids. And also, what you can observe is specific target binding, which can be modeled within this intracellular compartment. So in this example here, what you can see, again, is this drug Paclitaxel. And in this case, what we're looking at is the accumulation of that drug into tumor. And you can see, based on this figure, that there is a higher concentration that's observed in the tumor versus the plasma. So something also important to consider is that there is high variability in the clinical data. So what my colleague Siri was able to look at, looking at simulations of the intracellular and extracellular concentrations, what you were able to see is that there was significant intracellular concentrations which were highly sensitive to tubulin concentrations. And also those inter-individual differences in tubulin could affect some of the non-linearity that you could observe in this case, and affect accumulation of the drug. Finally, I just wanted to bring in an example and kind of a shout out to another colleague of ours at Certara, Tom Polasek, who's working in kind of a field of precision dosing. And essentially what this paper looked at, some of the group at Flinders, what they were looking at is using the Simcyp Oncology population to look at the sensitive parameters in terms of describing different covariates. So what they were able to look at in this case is they looked at the impact, for example, of 3A, and also the effect of albumin. And in this case, what they were able to show is that these covariates were both important for predicting and also driving variability in this compound's exposure. So just to summarize, I've just gone over some of the use of physiological data and how we can use that to build PBPK models for healthy individuals versus oncology patients. Looking at case examples using PBPK in oncology in terms of informing drug labeling decisions. I've given you a couple of examples looking at PD and also looking at modeling intracellular concentrations. And finally, I just wanted to show you, based on that recent publication, the application of the population to identify key covariate information. So just to sum up, just some acknowledgements when I close. My colleagues here at Simcyp and also consortium members, Genentech and Sanofi in particular, who were generous enough to share patient data in terms of allowing us to build those physiological parameters and verify our population. Okay.
Great, thank you.
Yeah, if you have any
go ahead, Ollie.
Just if you had any questions, yeah, I'm here.
Okay, wonderful. Thank you so much, Ollie. We've gotten quite a number of questions from our audience already. That's fantastic. We'll try and answer as many of them as we can. So, Ollie, the first question is, someone wanted to know about the how accurate is the renal clearance calculated using the Cockcroft-Gault equation?
So out of using the Cockcroft-Gault and modified diet and renal disease, it was reasonably okay, but it didn't capture all the physiological data that we had for GFR. So that's why we started looking at other equations in the literature. So that was why we recommend this right formula, because when we did all the verifications, this seemed to be the best model for predicting GFR in cancer patients.
Great. Another question someone wants to know. Sorry, just one sec, having a little bit of a trouble with the windows here. Is the change in plasma protein that you noticed, that you notice in all types of cancers? Is it specific to certain types of cancer, for example, liver cancer?
Sorry. I would expect specific changes in liver cancer, but the data that we had were for a large array of cancers. So this is a large meta-analysis of different types of cancer. So, and this seems to be the general trend. In terms of albumin as well, it's well known as kind of a prognostic marker of how severe the disease is. In terms of when that drops quite significantly, that can indicate quite severe cancer.
Have you looked at the prevalence of hepatic dysfunction in cancer patients?
Not specifically, and it is something that sometimes can go hand in hand. Yeah, we didn't kind of tease out those different effects, but it is something that can be seen in cancer patients.
Can you expand on the differences in transporters observed between healthy and cancer populations?
Yeah, sure. So if I go back to the slide, maybe that'll help. So sorry, this is a bit small, and I didn't have time to go through everything that we did on the kind of transporter side. Essentially what we did, so in 2016 we had a publication which included a large meta-analysis of transporter data from different groups. And essentially that was all combined into a database. And when we put that together, we had indications of kind of disease types and different other parameters that we were able to select data from. So this data was actually excluded from the healthy data set, obviously, but it became useful for looking at what differences that you might see between cancer patients and healthy individuals. It's worth pointing out that it's relatively small data set compared to the healthy individuals, but what was done was looking at significance in terms of significant effect versus in change of abundances versus healthy individuals. And in this case, what we looked at are changes in O2B1B1, which was observed to increase. There were decreases in 2B1, MATE1, OCT1, 1B3, and other transporters, hepatic transporters that we looked at didn't have any significant change versus healthy values. But the other thing to point out really is that this is based on in vitro analysis of samples. So liver samples, for example, have been taken and then measured primarily using kind of LC-MS approaches. And essentially, there isn't much in vivo data to properly verify these. So there is some caution applied, but we feel that there is enough evidence at the moment to justify putting that in with the population.
Ali, someone wants to know whether the Simcyp simulator has an option to add a metastases compartment in one of the organs, for example, metastasis in the liver.
So we don't at the moment have that facility to be within organ. Everything is kind of as an additional compartment. There is obviously significant interest in that, but this was kind of our first step in terms of doing that. There are obviously a lot more complications in terms of doing that within the PBPK model, but it's something that we would like to do eventually, yeah.
You may have answered some of this already. I know you talked about in vitro data that you collected through LC-MS, but one of the attendees wants to know, can you provide some examples of the in vitro data that was incorporated into the model?
So you mean for the transporter abundances, or so for the transporter abundances, the healthy values are published. I'm not sure what else you mean. In terms of the cancer population, we are in the process, as you saw with Malice's paper, in terms of looking at PD effects. We're publishing the cancer population alongside that, so the kind of data behind that population will also be published shortly.
Can this approach be used to predict the concentration of oncology drugs in both plasma and tumor at different stages of cancer?
Essentially, yeah. That's kind of the aim. You would have to look at the kind of physiological differences between those different stages. In terms of tumor, you can, as I mentioned previously, you can look at the growth of that tumor as well, so you can model that in terms of its change over time within a simulation. In terms of the individuals, in terms of their physiology, so changes in like plasma protein, for example, that would be kind of statically modeled, where you would model a particular time point of disease and then look at any other physiological changes that happen at different time points. But essentially, you could utilize the model to do that kind of approach.
Does the change in protein binding of drug seen in solid tumors, does that also occur for liquid tumors as well?
I'm not qualified enough to know all about liquid tumors, so I'd have to defer that question, but I can definitely find that out.
The 36% decrease in clearance doesn't necessarily mean that there's no change in CYPs, as other factors might play a role, such as changes in protein binding, transport expression activity, etc. Did you rely on in vitro activity and abundance studies of CYPs in cancer?
So that was an interesting kind of part of our research. So actually, some of the biggest data sets, or at least the old data sets, do seem to have a lot of cancer patients which were originally used for kind of building as a backbone in terms of the absolute abundances which were previously measured, for example, in Western blotting. So what we tried to do is tease out studies which specifically had separated out cancer patient abundances and healthy cancer abundances in terms of CYPs. And there weren't any real conclusions. I didn't have time to show the slide that we've prepared on that particular aspect. But essentially, in some studies, you could see that there might be a change. In other studies, you saw that there wasn't any real change. So that was relatively inconclusive in that regard. And some of the kind of examples that are listed there, actually what I wanted to highlight was that it was the change in intrinsic clearance of drug. So that was something that was particularly influenced by the abundance and also changes in liver volume, which are accounted for when we look at changes in age. So what you get is a decrease in that liver volume. And in this case, we felt that if we took into account age properly, that reduction, overall reduction, was seen just by taking into account age in this case.
A couple more questions. Can large molecules be modeled using the tumor model?
Yes, yeah. So I didn't have time to cover this, but essentially you can look at other proteins and peptides, as well as monoclonal antibodies and even ADCs. So between those two, there are two slightly different models which are used, but essentially you can model a tumor compartment for any of those, and you can look at target-mediated drug disposition as well. So those factors can be taken into account and used to model for large proteins as well.
Looks to be the last question. Is there a pediatric cancer population in the Simcyp simulator?
Right. So at the moment, no, we don't have a pediatric one. What you can see in two of the examples, one of the examples I showed actually, they looked at this kind of bridging for docetaxel, and a kind of similar approach has been applied by one of my colleagues Jennifer Bonner when she was at the University of Newcastle. And essentially they were looking at actomyosin D, and again, what they were looking at is pediatric dose bridging. One important thing to note is that in both cases, what they assumed is kind of the healthy pediatric physiology. And primarily that's because there isn't any real data sets which are available. They did start doing some work in the University of Newcastle in the UK, and essentially they looked at measuring different patients for example, plasma proteins. So they saw that there were significant increases in AAG, similar to what you would see in adult, but in terms of other parameters, so height and weight, they didn't see any real significant difference between healthy. So I guess the point is that it's not available. Some people have started looking at it, but partly it's availability of data which is required to drive that one forward as well.
Great. Thank you so much, Ollie, for that excellent presentation, and thank you so much to our audience for all the insightful and thoughtful questions that they asked.